Hi guys! We are from HP1 Group 2. Today we're going to conduct an experiment about DNA extraction and gel electrophoresis. Stay tuned!
First thing first, we are gonna smash a few frozen strawberries in a plastic ziploc as you can see here. Next, we're preparing the extraction liquid to ease the DNA extraction. First, we will need 5 spatulas of salt in a beaker. Then, we are pouring 250 ml of distilled water and 20 ml of this dishwashing liquid into the same beaker.
Now that the strawberries are ready, we're going to strain the mixture into a separate beaker until there is very little liquid left. Now that we have the strawberry juice in the beaker, we need to transfer it into a 15mm tube and very slowly 15 ml of cold rubbing ethanol is poured down the side of the tube.
In this part, we are going to build a comb structure using craft sticks and attach them together using tape. Make sure there are at least 4 wells between the teeth. In the meantime, we are gonna prepare the gel for the electrophoresis. First thing first, measure 2 grams of agar powder using an electronic balance into a beaker. Mix the powder with 100 ml of buffer solution containing distilled water and sodium bicarbonate. The mixture is transferred into a conical flask and is put into the microwave. No exact time is determined for this step, but we definitely need to heat it till it bubbles. Now we have it. Lastly, we are gonna pour the hot gel mixture into the container with our comb set straight out. We'll come back in a moment when the gel is cooled and hardened.
Since we require electricity to conduct this experiment, we're gonna need to cut some wires and bend them into these shapes. Remember the strawberry ethanol mixture that we had in the first part? Now it's making its comeback. The DNA sample is taken and mixed with 0.5 ml glycerin in a small tube.
Now that the gel is hardened, the comb is removed and we are going to put a few drops of food coloring into 3 out of 5 wells we have. They must be in different colors, so here we are using red, green and blue. Next, we will drop some methylene blue dye into one of the wells, and for the last well, we are going to put our strawberry DNA samples. Last but not least, we are going to connect our power supply to the submerged wire of the gel. Then wait for 15 to 20 minutes and
a few moments later.
Here's our final result. After we remove the power supply, we can observe that the food colorings have different length of bands as they travel across the gel at different speed. Not only that, we can also see that the methylene blue dye and the DNA sample are moving in opposite directions of the gel. This is because methylene blue is positively charged, which makes it travel to the negative pole, while the DNA of the strawberry are negatively charged as they travel to the positive pole. DNA gains its negative charge from the phosphate backbone, while methylene blue dye is cationic.
So, what are the conclusions of this experiment? The experiment is said to be successful as we can see all the fragments of dyes and DNA sample to be separated. Overall, the experiment demonstrated the fundamental principles of gel electrophoresis: separation, movement under electric fields, and molecular comparison. It confirmed that agar gel can serve as an accessible medium for visualizing molecule movement and that DNA samples from strawberries can be effectively separated and observed using simple, affordable materials.